on the WNT website afterwards. So if you want to discuss, you can use the chat box to ask questions or discuss, respond to questions. The WNT will have an opportunity for some follow-up as well, and a short report will be prepared by the Secretariat. So I think I'll stop there, and I'm going to hand over to our first speaker, checking with Henrik. Henrik, are you here?
I'm here, yes. Can you hear me?
Yes, very well, Henrik. Good afternoon.
Good afternoon.
Yeah. So I'm going to project your slides, Henrik. There we go. Okay, can everyone see the slides? Henrik, can you see your slides?
No. I can see the program for today.
Okay.
Now it comes. Yes, now I see it. Okay. Yeah, thank you very much, Anne, for inviting me to give a presentation here at this webinar. Now there'll be quite detailed presentations on the tools and approaches by the next speakers. So I'll give a more general introduction to the cross-species extrapolation and methods related to that for ecotoxicology here. And my name is Henrik Holbech from University of Southern Denmark, affiliate SDU and not DTU as written, but no problem. Next slide, please. Okay, we got a few topics to discuss, new approaches related to cross-species extrapolation in ecotoxicology, and then how they differ from the human health methods applied, and discuss some specific challenges that poses for validation or predictivity or standardization for the wildlife. So these are the three topics I will try to cover now. Next slide, please. First of all, cross-species extrapolation of toxic effects is, of course, not a new topic. It's been used for decades for the chemical safety assessment. We all know for human health about the mammalian testing, mostly with rodents and, for example, dogs, or the environmental toxicity testing with all the non-mammalian species from a number of different taxa. So it's for sure not a new topic, and it's been used for a very long time. Next slide, please. Discussing ecotoxicity, it's very different from human health because each of the standard test species are going to protect hundreds or thousands of species, especially when we discuss the invertebrates. And due to this, there is normally included uncertainty factors or safety factors to support or to protect the species differences. Next slide, please. And here's just an example from one of the European regulation REACH that are covering all the industrial chemicals. And here you see data for aquatic ecotoxicity. So it's all related to the number of informations or the type of information you have. And if you only have, for example, one short LC50 test, then you use a safety factor of 1,000. And if you have chronic tests with NOx, the more tests you have, the smaller a factor you can use, until the top of the information level, species sensitivity distributions. If they are very detailed, you can go down to having actually no safety factor. So this is the standard way of doing it, doing in vivo testing and adding assessment factors. Next slide, please. But there's a number of shortcomings with the strategy just mentioned. Of course, we would like to reduce animal testing. That is one of them. The other one is that we don't get enough information for all the different species. in the environment by using this approach. That's why we add the safety factors that are maybe safe, maybe not. We actually don't know for all species and classes. So therefore, we have are going to implement a number of new approaches. One of them is the NAMs, new approach methods covering in silico and in vitro assays, and also alternative animal species than the normal used. Then we have the IATA, part integrated approaches to testing and assessment, where we try to do chemical grouping and read across, defining testing strategies, and they are very often related to the AOPs, that are the adverse outcome pathways and also the AOP networks. I'm not going much into detail with AOPs because Dries will do this very shortly. And then I added also the considerations related to 3Rs with our in vivo experiments, existing in vivo experiments, where work is ongoing on adding more endpoints to the existing tests so we can get as much information out of in vivo testing as possible. And then my last point is the invertebrate. That is, I think, a special issue related to read across that I'll come a little bit more into details with later. Next slide, please. So just how do these different new approaches differ from the human health methods? Let's start with the NAMs. If we look into the test guidelines and in vitro assays, there is one for ecotoxicity, and that's the TG249 that was adopted last year, the rainbow trout gill cell line assay. And this is compared to much more than 20 human health in vitro assays or human health related in vitro assays. So there's quite a big difference in the in vitro assays at the moment. On the other hand, we have a number of embryo assays in ecotoxicity. Newly adopted ones are those mentioned here, 248, the CETA, and 250, the EC assay, and the last one is the radar 251. And then I forgot to write it in here. Of course, we have the fish embryo toxicity test 236 that is also regarded as a NAM. And these are very important in these new approaches to toxicity testing in sorry, in my point of view. Next, please. Then we have the IATAs. I checked in the OECD case studies. I think now it's more than 20 case studies. And next to one on bioaccumulation, I think all of them is focused on human health. So that's, of course, a different from the Ecotox part. I know there's a lot ongoing on acute fish toxicity and IATA within the BMD Eco. And so work is in progress, but the focus on the case studies until now has been more or less 100% on human health. So there we need much more emphasis in the coming period on the ecotoxicity. And the next, please. And then we have the AOPs. Fortunately, that's a good way to integrate human health and ecotox effect assessment by AOP networks. And I'll come a little bit more into this, but as said, Dries will comment on this in the next presentation. And the next one is the advancement of the existing in vivo test, where more endpoints could be included to elucidate more mechanisms and more modes of action. And at least for part of this, work is in progress. For example, thyroid hormone system disruption sensitive endpoints are on the work test guideline work program now, with the inclusion in the existing fish test guidelines. But of course, much more should be done on this on mechanisms not yet covered. And here I especially speak about the endocrine disruption part. Next slide, please. Invertebrates, covering more than 95% of all animal species. Here we, with the existing testing strategy and test guidelines, we are not just doing cross-species extrapolations, we are also doing cross-classes extrapolations. For example, echinoderms are not covered by the existing guidelines. And if we speak more specifically about endocrine disruption, then it's actually not even possible to identify a chemical as endocrine disruptor, according to the WHO definition, in any invertebrates, because we only have diagnostic endpoints, reproduction, and so on, but we don't have any specific endocrine sensitive endpoints. So there we have a lot of issues, and hopefully now we are going to hear John on PrecisionTox later on, that there will be some tools that can support to close some of these quite big gaps related to invertebrates. Next slide. And then we were asked to discuss also the specific challenges for validation, standardization, and predictivity for wildlife. And of course, we should recall that one of the difference between human health and the environment is that in the environment, adversity is mostly related to the population and not the individual. And here we can have some challenges, especially when we talk about non-standard endpoints for adversity. Next slide, please. One of these examples is from the fish and amphibian studies, where sex ratio is an adverse endpoint, but what about the size of the adversity and the biological relevance? I just want to show you here a modeling from 2014 by Hazelrig et al., showing what type of skewing of sex ratio should actually happen before you see a 10% reduction of the population size. And what you see in this picture is that if you skew a little bit toward the females, you'll actually have an increase up to 20% of the population. And this model is on zebrafish, and zebrafish are continuous spawners, so even the spawning strategy would change these modeling or these models very significant. So relating a change in a sex ratio to adversity can be really complicated, even just for a few fish species. So that is one of the challenges in the new approaches, also that hopefully can be supported by some of the methods we will hear about a little bit later. Next slide, please. And then next to the non-standard endpoints, there's also other effects that are challenging. Next, please. For example, using the mammalian endpoints, which we would really like to do much better. There's also mammals in the environment. But how do we translate a change in an genital distance in a rat to a population relevance? That is really difficult. Hopefully AOPs can support this. Maybe it is an indicator of change in sexual behavior that could be an adverse effect, and so on. So there's a lot of AOP work that we need to support, for example, read across from mammals to non-mammalian vertebrates. And then one thing that is very specific for Ecotox is that it can be very difficult to predict species and class-specific effects. I think one good example is tributyltin that causes this effect in gastropod marine snails, Imposex. I don't think any of the existing test guidelines would catch such an effect, especially not those that are in the general standard requirement for regulatory use. So hopefully again there, the new tools can go in and support these class and species specific effects. Next slide, please. Now, this is just an example of cross-classes extrapolation taken from one of the projects, Ergo. that both Dries and I are involved in, where we try to break down the wall between human health and the environment. And this is a quite complicated figure, but what is trying to do is using the thyroid system, which is very well conserved among vertebrate classes. And then based on existing information, we'll take chemicals with known molecular initiating event and key events and endocrine activity and adverse effect in mammals, so known from mammals, and then investigate this in silico in vitro and in vivo in fish and amphibians. And then try to develop a battery of biomarkers and endpoints and test them for endocrine activity and adverse effect to confirm that you see the same in vivo effects in the amphibian and or at least adverse effects in amphibians and fish. And then do some biotransformation data when you don't see the same activity and adversity, and take all these existing data and all the new data we are producing to support the AOP development and AOP network development especially. And then the other way around, we'll take chemicals where there's unknown effects in mammals, but with known activity and adversity in non-mammalian vertebrates, and look into in silico and in vitro differences in biotransformation among mammals and non-mammalian vertebrates. And overall, hopefully this approach will support read across between classes also, and not just between species. I think this was my last slide. Yeah, I know this was a very brief overview of some of the topics we discuss related to extrapolation between species. But hopefully it was a little bit helpful to support the next more detailed presentations. Thank you very much.
Thank you very much, Henrik. I think this was an excellent introduction to this webinar, and a good transition also to our next speaker, Dries Knapen from the University of Antwerp in Belgium. Dries, can you hear us?
I can hear you. Good. And I can see my slide.
Excellent. So please go ahead.
Thanks. And thanks, Anna, for inviting me. And thank you, Henrik, for providing this excellent introduction, which really set the stage for many of the topics, as you said, we will discussing later in this webinar. So for my presentation, I came up with the title of AOP network-based cross-species extrapolation, challenges and opportunities. And you'll see that we'll have many of both of these two. Next slide, please. So the background of my presentation is an initiative that is supported actually by a number of projects, but mainly by the ERGO project that Henrik already talked about as well, in which we aim to develop a cross-species AOP network for thyroid hormone system disruption, where we are hoping to add a number of endpoints that are covered by the key events in the network to existing test guidelines that would hopefully allow us in the future to use in a cross-species extrapolation context as well. So the conceptual network is depicted on the right-hand side here. And just by looking at that, I think that anybody can see and feel the potential and the opportunity for doing cross-species extrapolation based on AOP networks. Cross-species extrapolation always has been one of the great promises of AOPs and AOP networks. But actually the challenge really is to make it happen and to find out what the best approach is for doing so. So I want to emphasize that this is work in progress. This is still evolving. It's an evolving science as well. And today I just wanted to cover some of our past experiences and some lessons that we learned so far by trying to do this. So next slide, please. So as an introduction, I want to briefly introduce the concept of the biological domain of applicability of an AOP. So the biological domain of applicability is the domain to which an AOP applies in terms of sex, life stage, and taxonomic groups mainly. So it's not just taxonomy, there's other components to that as well. But today we'll be focusing mostly on the taxonomic part of that domain of applicability. There's an important distinction in describing the domain of applicability between the empirical domain of applicability, which is obviously based on data, versus the plausible domain of applicability, which is based on scientific reasoning, for example, molecular conservation of target or phylogenetic relationships between species or between taxonomic groups. And a very important component will be, when we think about this in terms of AOP networks, is the fact that the domain of applicability of an AOP as a whole is not necessarily the same as the individual domains of applicability of the individual key events and key event relationships that are part of the AOP. I'll explain that in the next slides. So next slide, please. So this is a brief example of a part of this larger AOP network for thyroid hormone system disruption dealing with fish. I'm just showing this as an example. This deals with two different molecular initiating events leading to effects on inflation of the swim bladder of the fish. And this is a small sub-network of the overall AOP network, and it already illustrates some of those concepts. For example, you can see that part of this network only applies to early life stages, while another part of the network only applies to later life stages. And as a whole, you could say that in terms of taxonomic applicability, this network only applies to fish species which have swim bladders that consist of two different chambers, because that's the model of the swim bladder that was used for developing this network. So that's how we think in terms of taxonomic domain of applicability of a network of AOPs. So next slide, please. But to assess the domain of applicability of an entire AOP, actually that's an effort that consists of different sub-elements. So basically the domain of applicability of the key events and the domain of applicability of the key event relationship. So the arrows connecting the boxes. These two things are separate efforts and should be done separately. So for example, you can see in this conceptual example that key event 2 is a key event that would apply to fish and amphibians, for example, and key event 3 would only apply to fish. That would mean that the key event relationship connecting these two key events, so the arrow between key events 2 and 3, would only apply to fish and not to amphibians, for example. And this has consequences for the domain of applicability of an AOP as a whole, which will nearly always be defined based on the most narrowly restricted domain of applicability of all of the key events and key event relationships that are part of that AOP. And I know that this sounds quite theoretical, but it has very important repercussions when we start building AOP networks. And the next two slides will illustrate that. So next slide, please. So this is one of the actual AOPs that are part of this larger thyroid AOP network, leading from thyroperoxidase inhibition to swim bladder inflation. And here I conceptually wanted to illustrate this idea, where the first part of the AOP, so the molecular initiating event leading to a decrease of hormone levels, is something that's more than likely applicable to many, if not all, vertebrate groups and classes, because the HPT axis is something that's highly conserved across groups. While obviously the part of the AOP that deals with inflation of the swim bladder is only applicable to fish, because only fish have swim bladders. But then going further down to the right, the swimming performance key event, for example, might be applicable not only to fish, but perhaps also to amphibians. And then the adverse outcomes at the population level, like increased mortality leading to decreased population trajectory, might be something that's even applicable not only to vertebrates, but also to invertebrates. So a typical AOP would have a relatively narrow, often not always obviously, but often would have a relatively narrow taxonomic domain of applicability, so abbreviated as TDOA in the middle part, and have a relatively broad TDOA to the left and to the right of the specific parts of the AOP. So this is relatively straightforward to understand from the perspective of a single AOP. But the real challenge arises when we start to develop networks based on such AOPs that connect to each other at different places of the AOP with different levels of taxonomic, well, different domains of applicability, basically. So next slide, please. So AOP networks are simply said connections of AOPs. So as soon as two or more AOPs share a key event, they form a network. And a very important and actually forming the major challenge in this whole cross-species extrapolation in the effort. An important element of that is the concept of emergent AOPs. So emergent AOPs are new AOPs that emerge from connecting existing user-defined AOPs, which is explained to the right. So there we have two AOPs which share one key event, and as soon as we connect them, a number of additional AOPs emerged from the network that have never been explicitly developed separately as AOPs by AOP developers. And so that's the real challenge of building AOP networks. So next slide. So in this slide, you can again see the conceptual overall AOP network for thyroid hormone system disruption across these different species. More or less, and I'll show in the next slide, I'll show a picture that's more detailed in a minute, but this more or less shows the current status of this network as it is in the wiki. Meaning that, for example, the adverse outcomes of hearing loss and kidney toxicity and cognitive dysfunction have only been developed so far as AOPs for mammals, which doesn't necessarily mean that obviously these do not apply to other species like fish or amphibians, etc. So that's part of the assessment of the taxonomic domain of applicability of the entire network, to really make sure that we understand the taxonomic domain of applicability of all the different components of the network. And basically that if a specific taxonomic group is not mentioned as part of the taxonomic domain of applicability, that it actually also means that the AOP or the key event does not apply to that group. And that's really challenging because right now, if you would go into the wiki and you would not find a taxonomic group mentioned, for example, it could also just be that people didn't write it there, but it doesn't necessarily mean that it doesn't apply to that group yet. And so that's what the assessment of the overall network entails, basically. And so in the next slide, you can see the actual network as it currently exists in the wiki. So this is the network for thyroid hormone system disruption based on the 50 user-defined AOPs that are currently in the AOP wiki. It consists of 13 MIEs and 9 adverse outcomes. And it applies to mammals, amphibians, fish, and birds to some extent as well. And so the colored bars underneath the key events indicate what is currently in the AOP wiki in terms of taxonomic information to which these key events would apply. But as I said a minute ago, obviously this does not mean that it doesn't apply to other species. Perhaps it just hasn't been described yet. So the real challenge is to come up with a strategy for approaching this. For example, do we start from the MIEs and move to the right in terms of thinking about the taxonomic domain of applicability? Or do we do it the other way around, starting from the adverse outcome and moving our way towards the left? Or do we start in the middle, etc.? Do we focus on all of these AOPs or just sets of AOPs? Are we going to group them or prioritize them? Because the real challenge, again, are these emergent AOPs. And you can see that we have 50 user-defined AOPs in this network, but that we have 439 emergent AOPs. So the total number of actual AOPs in this network is nearly 500. So that's a real challenge. And we had to think about a conceptual approach of defining the taxonomic domain of applicability of the overall network. So next slide, please. So we started with the MIEs, and I think Carlie will provide more information later on about the SeqAPASS tool. But one of the things we did there is use the SeqAPASS tool to assess structural conservation of the different targets, so different proteins and enzymes involved in these MIEs. And you can see an example of such an analysis in the graph here, which provides evidence for a biologically plausible taxonomic domain of applicability, which we then supplemented with literature review and empirical evidence that we could find of these key events or these MIEs actually occurring in these different species. And this was the first step that we did. So basically we started at the left, at the beginning. And then in the next slide, this is where we really were wondering how to proceed next in terms of assessing this. So we decided we wanted to continue with assessing the adverse outcomes of the network, but not the adverse outcomes as such, because obviously if you would think about an adverse outcome like kidney toxicity, if you don't consider it in the context of thyroid hormone system disruption, it's just too broad, and it's not really meaningful to assess the taxonomic domain of applicability of such an adverse outcome if it doesn't have a specific toxicological context. But as I said, in the network, we have almost 500 AOP, so that's too many to evaluate. So we needed some kind of abstraction layer to approach this, to simplify the network, basically, to allow us to assess the adverse outcome taxonomic domain of applicability in this specific toxicological context. So we generalized the AOPs, as you can see conceptually in this figure, and we tried to come up with generalized AOPs that placed these adverse outcomes in the context of the hub key event. So basically, we would go in the literature and try to look for data supporting kidney toxicity across different species that are related in one way or another to altered thyroid hormone levels, for example. And the detailed information that we would find, so which specific MIE was it, which specific key events were altered, this was basically then mapped in the overall AOP network. So we basically used the network as a framework for mapping all of the empirical evidence that we found. So we did assess the adverse outcomes but within this specific toxicological context. Next slide, please. And so this was an approach that was, on the one hand, very efficient because it implicitly led to the taxonomic domain of applicability assessment of not only the adverse outcomes, but also of the most important hub key events that are directly linked or connected to these adverse outcomes in the network. So actually, it was quite efficient to do it this way because the coverage in terms of the overall network increased relatively quickly. On the other hand, it also meant that in some cases, some additional intermediate key events were required to capture all of the information that we found in the literature. But here is one example for developmental neurotoxicity, where you could see that only for mammals, there was an AOP available in the wiki describing DNT as a consequence of thyroid hormone system disruption. But it's clear that we did find a lot of evidence in the literature that this AOP or this conceptual pathway is also applicable to fish, to birds, and to amphibians. So overall, it is a time-consuming and intense process, but it really resolves much of these gaps and knowledge gaps in the network. Next slide, please. So this basically summarizes our overall conceptual approach of how we are currently working on improving the taxonomic domain of applicability description of the network. So in the first 3 steps, we built the network and we connected it in the right way, and we made sure that the basic description was correct. And then we started with the assessment of the taxonomic domain of applicability of the MIEs and the AOs, as I just explained. And we are basically now moving towards the middle of the network, going through the hub key events to other key events that are connected to that, and slowly increasing the coverage that we have of the overall network. Along the way, as you can see in this roadmap, we have explicitly foreseen time points where we have some outreach activities where we get in touch with other AOP developers and make sure that what we do is added to the AOP wiki, but also really importantly, of course, make sure that we are aware of all of these AOPs that are part of the OECD's AOP development work plan, and make sure that any changes that we apply are communicated correspondingly as well. But still, again, the total number of AOPs in the network is large, so we will still need a strategy for grouping and prioritizing AOPs or adverse outcomes, for example, that are of importance, of regulatory concern, for example, or that should have focus first in terms of developing this further, because it's quite clear that the overall effort of doing this for the entire network is something that will take really a long time. And an important final consideration in that context, and now I'm coming back to what Henrik also mentioned, is that we want to, in the end, use some of these assays that measuring these key events in the network to add to existing test guidelines, which would then be useful for implementing and applying this cross-species extrapolation strategy. So on the next slide, I am giving one example of how we are approaching that. So this is a project of the OECD's test guidelines work plan, so project 264, which is on the inclusion of thyroid endpoints to existing OECD fish test guidelines, including some of the embryo test guidelines that Henrik mentioned in his presentation. So, for example, we are considering to add the measurement of swim bladder inflation and eye development and hormone levels, etc., to existing fish test guidelines. And importantly, these assays and these measurements will be supported by AOPs, including the description of the taxonomic domain of applicability of these AOPs, meaning that eventually we hope that these fish test guidelines will not only serve the purpose of assessing toxicity in fish, but also that people would be able to go to the wiki to understand and read the taxonomic domain of applicability of the fish assays that they are using and trying to understand what that could mean in terms of predicting effects in other species than fish as well, based on that same data. So on my last slide, I summarize. Next slide, please. Yes, I summarize this idea, basically where we are trying to build the bridge between AOP development at the level of the OECD in the AOP development work plan on the one hand and the test guidelines work plan on the other hand, and really import assays that are supported by AOPs into existing test guidelines. Importantly, we consider this as a living initiative, and we consider these projects as living projects. So we will continue to add AOPs to and key events to the network. We will continue to add or suggest at least the addition of new endpoints to test guidelines. But importantly, we will continue to improve the taxonomic domain of applicability descriptions of these key events and these AOPs as well, which means that over time and as this effort evolves, that the cross-species extrapolation potential, for example, in this case of using fish data for extrapolating to other species, will also increase over time. So that's, in a nutshell, the overall approach that we have been working on for quite some years now. I think that was my last slide, so thanks.
Thank you so much, Dries. I think you provided an excellent illustration of the utility of the AOP development program. So we will move to our next speaker for today, Jessica Ewald. And Jessica, yeah, I have to stop sharing. And normally you have the possibility to get the, yes, excellent.
Okay, you can see?
We have the presenter right, we see your screen, Jessica, so please go ahead, thanks.
All right, so the next 3 presentations, including my own, are all related to toxicogenomics data. So I'm going to give more of a general overview of toxicogenomics data, various applications, and then some tools that I've developed for analyzing toxicogenomics data. And then the next 2 presentations will go into the cross-species bit in a bit more detail. So just for some very brief overview context of where toxicogenomics data fits in this effort to replace traditional toxicity tests where we're conducting in vivo exposures and measuring organism level outcomes, we're trying to develop these alternative tests that conduct, for example, mainly in vitro exposures, measure molecular level outcomes, which are then used to predict organism level outcomes. So, oops. That's the big picture where toxicogenomics data fits in. Toxicogenomics data are these technologies for generating comprehensive measurements of molecular outcomes. So this is simply omics data measured in exposed samples. It could be transcriptomics, so gene expression with microarray technology or RNA sequencing, metabolomics data, proteomics data, and then there's just this growing list of omics types. There are many potential applications of toxicogenomics data. Kind of the overall big goal that I alluded to earlier is trying to build some sort of predictive models that can be used to predict effects in whole organisms from in vitro tests. However, this is quite difficult. So there's some more targeted applications as well, such as general exposure biomarker discovery, transcriptomic dose response analysis. So this is where we try to identify the concentration at which there's a concerted change in gene expression, or you could do this with other omics types as well. Many people try to come up with some sort of reduced ohm. So this with gene expression would be a reduced transcriptome, which is just simply a smaller limited group of genes that is representative of some larger biological space that can be combined with specialized technology for high throughput applications. And then in the most general sense, all of these methods and then just toxicogenomics data in general promise to provide more insight into the mechanism and mode of action because the data is such high resolution and has so many details. So how are toxicogenomics data actually being used in risk assessment? There are a lot of interesting methods being developed in the academic literature, for example, combining machine learning with toxicogenomics data. However, how are these data actually being used in a regulatory setting? There is a useful study conducted a couple of years ago, just as an example, where they did a survey of all of the toxicogenomic applications in risk assessment at Health Canada. And they found that, well, there's a lot of talk of all these different methods in the literature. The actual uses were primarily weight of evidence approaches for mode of action characterization. So this is kind of just a standard analysis where we identify the genes and pathways or metabolites or proteins that are impacted by chemical exposure and then use them to better understand a chemical's mode of action. So this is exciting. However, these applications so far have failed to live up to early promises of how toxicogenomics data would completely transform the chemical risk assessment process. And there's still a lot of progress to be made and exciting things to come. However, we found there are a lot of challenges related to validation, standardization of the methods, and then also the complexity of data analysis and interpretation. So that's more what my work is focused on. So why is it so challenging? One reason is that even for this most general sense, just for using toxicogenomics data for mode of action characterization, it requires expertise in diverse areas. So there's the bioinformatics analysis itself, which for a typical toxicogenomics data set involves raw data processing of the large raw files, downstream statistical analysis and visualization, and this must be conducted for like tens of thousands of different outcomes. So that can get quite complicated. And then finally, functional analysis of the results. So integration of the results with various databases of information on the individual genes, metabolites, and proteins. All of this typically produces tables like this. So for example, this is a table of KEGG pathways that were significantly impacted by chemical exposure. And there's a lot of detail here, right? Specific pathway names, and then all these p-values and other statistics. And so to really make use of this, one needs to have a very deep understanding of molecular biology, as well as a really good understanding of different mechanisms of toxicity to understand how these things kind of link together. And then finally, to actually apply them, one needs to be familiar with the very specific decision-making context in which it's being applied. And these vary across organizations and countries. And so bringing all of these three things together can be quite difficult. So a question that my group has had was, can we try and help improve this with well-designed software? So can we really address the barriers to the bioinformatics analysis piece of this and a little bit with the interpretation? So the SeqAPASS lab at McGill University develops web tools for bioinformatics analysis. Pictured here are some of their very general, more popular ones. These are not designed specifically for toxicology, but for just general biomedical research. They've been developed for the last 15 years, and they're currently processing over 3 million analysis jobs per year, so they're quite popular. So the goal of my work over the last number of years was to adapt these platforms specifically for environmental toxicology. So that means providing annotation resources for the ecological species that are typically used in environmental toxicology, and then also integrating toxicology-specific databases for interpretation of the results. So the overall objective of this is to make toxicogenomics analysis and interpretation more accessible to more people. So there is a lot of software that could be relevant to environmental toxicology. So I'm just going to give a brief overview here before looking at one in more detail. So we have Ecotoxicology Analyst for raw RNA-seq reads processing for non-model organisms. We have Express Analyst, Metaboanalyst, and Microbiome Analyst for general omics analysis. So this is for generating these lists of perturbed features and then mapping them to their pathways. Some toxicogenomics specific platforms are FastBMD for dose response analysis and Ecotox Explorer for Ecotox chip data analysis. And then a newer one is EcoOmics DB, which is designed for cross-species insights specifically for RNA sequencing data. So now I'm just going to go through Ecotox Explorer in a little bit more detail because it's kind of the most comprehensive example of a toxicogenomic software. So Ecotox Explorer is this web tool for analyzing Ecotox chip data. The Ecotox chips are developed by the Ecotox chip project team. And they were motivated by the fact that RNA sequencing data, while it's extremely useful, is beyond the capacity for some research programs. It can be expensive, has complex data analysis, and there can be long timelines for acquiring sequencing data. So the solution to this was to develop some qPCR arrays that have 377 genes on them, with qPCR technology being more readily available to more labs. These genes were specifically chosen for mode of action and dose response analysis by kind of literature review and analyzing a whole bunch of RNA sequencing data to choose genes. The chips have already been developed for 3 model species, commonly used in environmental toxicology, and they're just about done being developed for 3 more specific ecologically relevant species that are relevant to the Canadian context. And then Ecotox Explorer is the web tool for analyzing all of this data. So in Ecotox Explorer, there are 6 main steps. The first is data upload, where CSV files that are generated by the qPCR machine are just dragged into the web tool. Next, the software runs the user through a series of steps related to quality check and normalization. Next, you perform differential expression analysis to identify the genes that are up and down regulated in that particular data set. Each of the genes is organized into these higher level Ecotox module gene sets that are designed to give an overview of the major biological processes that are impacted by chemical exposure. Next, there are a series of open-ended visual analytics tools. So these are a bunch of graphics that are fully interactive. So you can click on the various parts to bring up more detail. And it's a really good way of linking the big picture down to specific pathways, down to individual genes. And since it's all interactive, you avoid these massive tables that are really dense and hard to get into. And then finally, there's a report generation step where you can generate a PDF report of all of the different methods and parameters that were used throughout the analysis. So this can be helpful for reproducibility. In addition, I'd just like to highlight a few more features in the tool. First of all, we took care to go through the whole AOP wiki and try to cross-reference genes that were on the chip with genes that were in various AOPs and key events. And so now when you have your table of differentially expressed genes, you can just click a link beside the gene and it should pull up the relevant page. Also, we have dose response analysis in here as well, so you can perform this with the Ecotox chips. And then the final thing I'd like to say is that we tried to carefully design this workflow so that it would be the same no matter which of the 6 Ecotox chip species you're analyzing data from. So we tried to make the Ecotox modules as standard across species as possible. Although that's not always 100% possible because of just differences in the species groups. So one unique aspect of developing Ecotox Explorer was the fact that we were, over the last 4 years, continually testing the tool with the wider Ecotox chip project team. So this is 169 people in academia, industry, and government who have a wide range of bioinformatics experience. So this has really helped us to try and tailor the design towards people who haven't analyzed this data before. The Ecotox chip project is currently in the final stages, and more than 30 case studies with the Ecotox chips and the web tool are currently underway. These encompass a wide range of chemicals, involve cross-species comparisons, in vitro to in vivo comparisons, dose response analysis, cross-platform comparisons, and then multi-lab replication. And some recent feedback from all of this work is that undergraduate summer students were able to analyze their Ecotox chip data sets with only about 1 hour of training. So we think that we're really meeting our goal of making toxicogenomics analysis more accessible. So for some conclusions, I'd just like to highlight some strengths of the web-based toxicogenomics software. The main one is that with this software, we're able to empower toxicologists to analyze their own toxicogenomics data, which hopefully makes it more appealing for people to use and to collect. There's the strength to the ability to standardize workflows. So since it's all online and on a website, you can make the same workflow accessible to an unlimited number of people anywhere in the world. And then finally, we've found that these web tools are actually great for toxicogenomics education. Since they're so easy to just pull up on the fly just by going to a web browser, it's a great way to get people who haven't interacted with toxicogenomics data before, whether those are in a regulatory setting or in, for example, an undergraduate toxicology class. You can get people immediately trying out different workflows and playing with the data. So next, I just wanted to end with a couple notes on environmental toxicogenomics specifically. There are a lot of similarities with human toxicogenomics. So the actual statistical methods are very similar, but it is more challenging. And the main reason is a lack of bioinformatics resources. So especially compared to mammalian models, there's just like orders of magnitude less information out there when it comes to annotating genes and metabolites and proteins. However, toxicogenomics data also has this huge opportunity for providing information for cross-species extrapolation. Since DNA is largely conserved across species and we often find the same metabolites, this could provide a more of a biological basis for mode of action extrapolation. And so this is going to be more of the topic of the next two presentations. All right, so that's all I have.
Thank you so much, Jessica, for a refreshing presentation on the utility of toxicogenomics data. With you, it looks so simple. Hopefully, you highlighted a few challenges, including on the standardization aspect, and thank you so much. Again, please make use of the chat box if you have questions for any of the speakers today, or if you want to exchange views. I changed the features for the chat box, so everyone should be now able to post messages for everyone. So our next speaker is now John Colbourne, from the University of Birmingham. Hello, John. I guess, yeah, I just need to share. And here we go. The floor is yours, John.
Salut, Anne. Merci, and thanks to the members of the steering group for this workshop and this kind introduction. So I have the privilege of coordinating the PrecisionTox project. This project involves investigators from 15 organizations across 8 countries, and it's also supported by a 20 million euro grant from the European Commission's Horizon 2020 program. There's been a significant investment of the European Commission into new approach methodologies, and this project forms a cluster with 2 other projects, including Risk Hunter and OnTox, that altogether represent a 60 million euro investment in exploring the uses of new approach methodologies, primarily for human toxicology. But building off the short talks of my colleagues earlier, this topic of my presentation is about bringing and bridging a 20th century artificial division, especially among the 2 disciplines of human toxicology and ecotoxicology. And these ought to be united because there are many opportunities that now exist. These opportunities include technological breakthroughs, such as the ones that Jessica mentioned, fresh ways of thinking by our early career environmental scientists who are advocating for environmental health. And also there's the desire to revisit the common usage of animal testing in toxicology for detecting and, when appropriate, regulating hazardous chemicals. Next slide, please, Anne. Let's take, for example, the realization that predicting both human health outcomes and ecological hazards for the regulation of industrial chemicals is extremely challenging. And we've seen this from Henrik's and Dries's talks. And in part, this is because investigations of how chemicals perturb biological processes and that produces harm has historically relied on different animals on which toxicity can most conveniently be studied. Now, this dilemma has regrettably created an unnatural divide in the scientific criteria that sets the limits for humans and for the permissible levels of contaminants in the environment, and this for many thousands of synthetic compounds. Now, this division has been manifested by a questionable reliance on mice and rats as human surrogates, and this is by virtue of our shared mammalian biology, and also a reliance on a fish, a cladoceran zooplankton like Daphnia, and an algal species forming an aquatic food chain, and these are used as sentinels for setting regulatory limits to protect, in this case, environmental health, or in the case of mammalians, to protect human health. Now, adding to this dilemma are significant concerns that there's really no single model as a perfect human surrogate or as an environmental sentinel species, and that the traditional approaches that have been used so far, they offer little guidance for converting these test outcomes to evidence-based interventions that protect human health and the environment. So next slide, please. Now, fortunately, there is a solution here that's provided by a truly unified theory in biology. This picture I found and is very interesting. This is a picture of Theodosius Dobzhansky. And he asserted in 1973, in defense of the teaching of evolution in US public education, that, quote, nothing in biology makes sense except in the light of evolution. Now, Dobzhansky here is a central figure in the history of biology by having integrated genetics with evolutionary biology. And he has even helped establish the fruit fly Drosophila melanogaster as a model organism for experimental research, including research into the genetic causes of human disease. Now, his famous quote here has been repeated thousands of times in thousands of publications, and they're repeated to argue the centrality of evolutionary thinking in biology. Now, this includes a pivotal report that was published by the US National Academy of Sciences in 1998, which bluntly asserts that evolution, quote, get this, is the most important concept in modern biology, a concept essential to understand key aspects of living things. So my argument here is, well, why not understand key aspects of toxicology? Next slide, please. Now, if we're prepared to accept the centrality of evolution, to understand shared aspects of living organisms, then I'd like to postulate here that this is a key to integrating human toxicology and environmental toxicology, and this union for classifying chemicals based on pathways of toxicity that are shared among distantly related species by our genetic legacy. These have some epic contributions to make at understanding key aspects of disease biology and also at understanding the useful biomarkers of exposure-related adverse outcomes. Next, please. Now, thanks to Dries, we've actually seen now that more recently frameworks such as the adverse outcome pathway, they've begun to view and classify toxicity more in light of a mechanistic process than in the light of observed apical endpoints such as death or reproductive impairment. Now, in the case of adverse outcome pathways, Dries has pointed out that there are many components that form networks, and the components that he referred to here are these key events that come together in key event relationships that may be tied to adversity at different levels of biological organization. What's nice about these adverse outcome pathways is that they're chemical agnostic, but they're also species agnostic. We can very much apply them specifically to talk about any species, including people, but even extrapolate it to actually public health and the protection of ecosystems. It's worth noting that the adverse outcome pathway, which has been adopted by the toxicological community at large, is in fact a framework that was invented by an ecotoxicologist. Now, for the project of PrecisionTox, what I'd like to point our attention to is what's in this red box here. Certain key events occur early in pathogenesis and are molecular in nature. And these molecular key events also form networks of molecular key events that allow us to come up with a network of interacting genes and their metabolic products that is a necessary element of an AOP critical to the outcome. And it's these molecular key events that we're quite interested in discovering and applying as biomarkers to make cross-species predictions of hazards due to exposure. Next slide, please. Now, a central aim of PrecisionTox is to demonstrate, using data-rich approaches like what was presented earlier, and to explore to what degree we can discover these molecular key events that are conserved among major groups of the animal phylogeny, and that could be used to detect, classify, and potentially even regulate hazardous compounds. Now, in our project, we are choosing and testing 250 chemicals for their toxicity on 5 biomedical model species, including model species Daphnia, which is an Ecotox model species, plus flavored with human cell lines. Now, in this case, what we're trying to do here is that we are testing systematically the responses of chemicals on the phenotypic outcomes of exposure in these model species, but also extrapolating molecular key events from the application of both transcriptomics and metabolomics. And we're doing this in order to provide 21,000 samples that are then analyzed for changes in these gene expressions and changes in the magnitude of metabolites as a consequence of chemical exposure. Now, data science here is key because it will reconstruct these multi-omics responses of these model species, revealing the molecular key events of toxicity-relevant networks. Now, most importantly, the evolutionary principles that we talked about earlier are used to map the origins of these molecular key events on the branches of the animal phylogeny. So therefore, risk managers and chemical regulators, they will receive knowledge on the likelihood that humans can be harmed by classes of chemicals because we humans possess the same toxicity pathways as other animal species by evolutionary descent. But our approach here has the added advantage of detecting toxicity that is specific to any particular branch of the animal biodiversity, such as insects, for instance, that threaten food security, and even be able to predict no toxic effect in other species by detecting the lack of these molecular key events or the lack of these toxicity pathways in specific branches of the animal phylogeny. Next slide, please. So here I want to provide a good example of the value of seeing toxicity in light of evolution. And this was done from a literature-based investigation of a master's student of mine that I supervised last year. His name is Jamie Wall, and he had questioned a fundamental assumption about a class of chemicals made by the pesticide industry that is designed to inhibit chitin synthesis, so to be harmful to insects' growth and development, yet supposedly safe to vertebrates because vertebrates do not possess a chitin exoskeleton like Daphnia, like other like insects, which is the product of this complex biosynthetic pathway. Now, at first, this assumption by the pesticide industry seems rather logical because it requires a qualitative difference between the biosynthetic pathways of arthropods, which are the insects and the crustacea, which all contain chitin exoskeletons compared to vertebrates that do not. But Jamie's search here in the scientific literature revealed most recently experimental results obtained only since 2016, pointing to chitin synthesis, the synthase, the enzyme that's circled and colored in red here. This has been the primary target of these benzoylurea class of insecticides. And so therefore, this is the mode of action for this class of insecticides. Next slide, please. Now, this search that he's done on the sequenced genomes of all of these model species for genes that encode enzymes for this metabolic pathway invariably shows that this pathway is actually highly conserved among both invertebrates and vertebrates, except for the loss of chitin synthase in mammals, but not in fish, nor in non-mammalian tetrapods. And this fact is currently unaccounted for in the chemical regulation. So let's revisit what he had discovered here. It was assumed that this pathway to toxicity had originated at the ancestral branch leading to the zooplankton and the fruit fly here. However, what his investigation showed is that that pathway originates very deep in the animal phylogeny, at the base of the animal phylogeny, shared among all other species, but that the chitin synthase, that gene that is actually the target of these class of insecticide, was lost, subsequently lost in the early ancestors to mammals, including humans. And this was confirmed by experiments showing that chitin is in fact synthesized in the inner lining of the gut wall for developing zebrafish, and also found at the extremities of the hands and feet of the clogged frog. So these may actually be safe for humans, but they're clearly not necessarily safe for vertebrates. Next slide, please. Now, if we ended up investigating the conservation of all the genes across all animal species, and looking at what fraction of these known pathways or networks in humans are shared with other vertebrates and shared with distantly related invertebrates, we can come up with a predictive model for the pathway conservation. And we're talking not here of just toxicity pathways, but all pathways that are shared among animal species. And so this general linear model indicates that there is a function with regards to the general conservation of pathways that is dictated by the size of the biomolecular network. So if we look at this graph, for instance, the larger the human network is, and in this case, we're looking at human networks of up to 600 genes, the more we find them to be conserved among other vertebrates, including mouse, and that they're equally shared with the distantly related invertebrates, including Daphnia. So if we look at, for instance, where number 1 is located, we see that at very large human networks, we can have up to 100% conservation between humans and other vertebrates. But in this case, when we observe 80% conservation between humans and other vertebrates, we also see 80% conservation with the invertebrates. And vertebrates and invertebrates last shared a common ancestor back to the Cambrian explosion almost 600 million years ago. So this is a phenomenal finding. But this holds for network sizes of roughly around 300 genes. But as soon as you have smaller networks, these are co-expressed or co-associating genes for specific pathways. When they become smaller, you get to basically see a deterioration of the conservation between vertebrates and invertebrates. And therefore, we can actually make predictions here as to which toxicity pathways would actually be predicted to be observable in invertebrates, yet relevant to vertebrates, including humans. Next slide, please. So what does this mean? Well, the whole aspect of this investigation is to see to what degree we can make toxicological observations where the findings are broadly applicable across animal species. And what this effectively can do, which I call toxicity by descent, is to basically bridge that artificial divide between protecting ecosystem health and protecting human health. Because a sentinel or species such as Daphnia, which is sampled from the actual environment in which it lives in, can actually provide information as to whether these toxicity pathways are elicited by not only synthetic industrial compounds, but also by direct exposure of substances taken directly out of the environment. So toxicity pathway and the use of toxicity by descent is really an approach here of improving the protection of human health and the environment by assessing the substance's modes of action that are conserved biomolecular response pathways to toxicity. Next slide, please. Now, this really opens up the door here for what I call precision environmental health assessment. Because in all cases, what we're doing is that we're using data-rich approaches, such as transcriptomics and metabolomics, to look for the biological profiles or responses after exposure of sentinel species, such as Daphnia other invertebrates or the embryos of fish or tetrapods, and to basically drill down and to discover to what degree the responses here are predictive of adversity in other species. But now if you add a third large data set, and this is really non-targeted analytical chemistry of the environment, one could actually apply a three-tiered approach where you discover what components of all the mixtures that are found within environmental samples are the drivers behind the biomolecular responses that are observed in our sentinel species, and therefore be able to actually not only discover the molecular key events that are predictive of adversity, but also to distinguish those elements within a chemical mixture that are coalescing in their activity to drive that toxicological response. And so this is one place that maybe in the short to medium term, we can be in a position to apply these approaches that are being talked about here during this webinar into doing true environmental health assessment for both humans and ecosystem functions. Last slide, please. So there is potential improvements here in toxicity testing, and these are likely, and they're likely to be soon. The technology is mature. And so what we're looking to achieve here is some merging of the science with governance in order to provide adequate regulatory approaches to these problems. And so we're looking for a more robust comparative biological basis for assessing health effects of environmental agents using mechanistic data to provide a broad coverage of chemicals, chemical mixtures, the outcomes, the species, and life stages. And this, of course, is helped a lot by the AOP framework, and to reduce the cost and time of doing testing. And finally, to embrace this data-rich new approach methodologies to assess chemical modes of action and to act upon that knowledge. That's all I have for us today. And
thank you so much, John, for this insightful presentation and taking us through the phylogeny journey. Conscious of time, I'll move straight to our next and last presenter for today, Carlie Lalone. Carlie, you should be able to share your slides from your screen.
All right, I will do that.
Yeah, you're in presenter mode. Okay, excellent.
All right, great. Thank you so much, Anne, for having the opportunity to come and chat with everyone today, and for the nice presentations that I think really set up the talk that I'll be describing today, and coming from it from maybe even a different perspective than my academic colleagues, in that I've come from the Office of Research and Development at the U.S. Environmental Protection Agency. And much of our work is being able to really simplify these very complex concepts and utilize this type of information, evolutionary biology, bioinformatics, all these different types of approaches to be able to translate that science for our decision makers. And specifically working hand in hand with decision makers to be able to understand the types of information that is necessary for them to be able to utilize in their day-to-day pipelines. And so first, I'd like to just start off thinking about species extrapolation and thinking about the types of information that we're bringing to bear. So even when we're looking to move towards these new approach methods, such as high throughput screening assays, high throughput transcriptomics, and in the utilization of the adverse outcome pathway framework to understand biological pathways, many times we're still challenged with the specific organisms that are used as model species to be able to extrapolate that knowledge. Even in the context of AOPs, we may be defining an adverse outcome pathway with a single species or a handful of species. So we need to be able to think about how we can use those surrogate data from specific organisms and extrapolate that knowledge to other species. And as it's been discussed, we have a subset of model organisms that are utilized, and many times they weren't selected because they were the best representative species of a specific taxonomic group or across taxonomic groups, but typically because of their ease of maintenance or breeding capabilities or the ability to control their environment makes them a very useful model organism or species. So what we're trying to do with species extrapolation is take this knowledge about a single species or a handful of species and really predict or project the trajectory on other species. And in the context of chemical safety assessments, we're thinking about toxicity and how that can be extrapolated to other organisms. So there's many different tools that can be applied for species extrapolation, but one that I want to focus on today is the use of bioinformatics. And I think Jessica did a really nice job of talking about toxicogenomics and how we can utilize mathematics, information science, as well as biology to answer these large biological questions and explore very large volumes of biological data relatively rapidly. And so the use of bioinformatics really allows the computers to do the work, to be able to take advantage of information on, say, DNA, RNA, and amino acid sequences that lead to proteins, for example, to allow us to ask very complex questions and then process data relatively quickly. So, in starting to think about species extrapolation, we can begin to understand similarities and differences between species by looking at, by looking at specifically existing information. So with proteins, and actually we were talking quite a bit about genes, there's expanding databases because sequencing technologies have become so much more efficient and improved. So this is just an example from the National Center for Biotechnology Information, where the protein database itself is over 1 billion proteins representing, over 2 million organisms. So as these types of sequence repositories expand, and as Jessica had mentioned as well, bringing in new species and more information across a number of different organisms that are non-mammalian species and invertebrate species, we can really capitalize on that existing information to utilize for species extrapolation. And that's exactly why the US Environmental Protection Agency developed the sequence alignment to predict across species susceptibility tool to be able to take that knowledge and simplify a means to be able to transparently extrapolate information from single species or a handful of species to the diversity of species in the environment by using some relatively simple, but yet, bringing together a number of different tools to put these approaches in the hands of not just researchers, but also decision makers. And so SeqAPASS was originally released to the public in 2016. So it's a publicly accessible web-based tool that allows us to, ask a pretty simple question that really focuses on that chemical protein interaction, or it can focus on a protein-protein interaction. But there's a couple of pieces of information that we need to have to be able to begin a SeqAPASS query. So we wanted to simplify the utility of the tool by asking what is our knowledge about that chemical protein interaction? Do we have a known sensitive species or a targeted species for a particular chemical? So this would be in the context of, a pesticide. We may know specific species that are the target organism. We could use that as our query species in SeqAPASS. Or for, say, pharmaceuticals, you would think about humans or companion animals for which that drug has been designed. Also, in the context of high throughput screening or high throughput transcriptomics, we may have knowledge of a model organism used in an assay. So in an in vitro assay, for example, so that would be our starting point or our starting species using that model organism and the gene that was used in that assay. And then as Dries also spoke about, is in the context of the adverse outcome pathway framework, we may have identified an individual species that's typical within the AOP wiki, or a handful of species for which the molecular initiating event or early key events have been defined. And that species and protein interaction could be the initial starting point for the SeqAPASS query. So bottom line is, once we have that protein of interest, so that molecular target for which a chemical would act upon, or the protein which is involved in an adverse outcome pathway element, we can take that protein and species, and we can take the chemicals, molecular target in that targeted species, and compare it to the millions, billions of proteins from thousands of species. And what we assume then as that the greater the similarity between those proteins, the greater likelihood that the chemical could then interact in the other species. So what this does is it gives us a line of evidence for conservation of a particular target if we're thinking about it in the context of an adverse outcome pathway, or it gives us, again, the evidence for predicting potential chemical susceptibility across species, looking at conservation. So we designed the SeqAPASS tool to be very flexible in the analyses. There's 3 levels to the SeqAPASS tool. So you can be very strategic based on the information that exists on a particular chemical. So we can look at the full primary amino acid sequence, comparisons across species. If there's knowledge about functional domains, so such as in a nuclear receptor, a ligand binding domain or a DNA binding domain, we can look specifically at those regions for conservation. And then when we have knowledge of how specifically a chemical interacts with a protein, so maybe from X-ray crystallography knowledge or from site-directed mutagenesis studies, or even from mutations that occur naturally in the environment for making certain species less susceptible, say, to a particular pesticide, we can utilize that knowledge to look at those changes in amino acids across species, all along gathering lines of evidence towards protein conservation. And I won't have time to get into this today, but we are moving more towards structural evaluations and looking at how we can utilize protein structures for comparisons as well. So, in thinking about the SeqAPASS query and what the information that is generated looks like, we have a prediction of susceptible yes or susceptible no based on the lines of evidence towards conservation. So we really end up with a yes or a no prediction of susceptibility or conservation of that particular protein in another species. And again, we can start with any species, and the results look at the diversity of species, giving yes or no prediction. So this is something that was requested from program office partners within the EPA as we were developing the tool. So I think I have to be very clear about the SeqAPASS tool and what specifically we're predicting. So we're really asking a very simple question of whether or not a specific protein target is available in another species for that chemical to act upon or not, or is it conserved in another species? And so we have to be clear that there's many factors that make up, how a species is sensitive, including the exposure, dose, ADME, life stage, life history, et cetera. So this is a very important piece of the puzzle, but it's a line of evidence towards understanding whether or not a particular species or group of species is likely to be susceptible to a particular chemical, or in the context of the adverse outcome pathway framework, conserved across species. So some of the strengths of the SeqAPASS tool is that it is publicly available. It's very rapid. You get lines of evidence towards conservation for hundreds of thousands of species very rapidly. And it takes advantage of extremely well-established tools and databases from the National Center for Biotechnology Information databases that are, they utilize information from various other resources that are established across the globe as well. And what this provides is a very streamlined, consistent, and transparent method for generating information for extrapolation. We've published many different case examples, and we continue to do that whenever we add new features to the SeqAPASS tool, as well as providing user guides. One of the things that we do is we look ahead as bioinformatics approaches become more user-friendly, and we recognize that they can be utilized for automation or semi-automation, such as what I had mentioned, looking at structural evaluations in homology modeling and molecular docking, virtual screening. Those are the types of approaches that we would like to continue to evolve into the SeqAPASS tool as they become readily available. So as we develop the features of the SeqAPASS tool, we want to demonstrate application of the tool towards where there's specific challenges in environmental toxicology that are brought to us through our agency and through regulatory decision makers. So some of the applications that we've looked at for the SeqAPASS tool and demonstrating its utility are listed here, centered around extrapolating high throughput screening information. If we think about the endocrine disrupting chemicals, we looked at endocrine targets and extrapolating knowledge from ToxCast, for example, across species. We use the tool specifically to predict relative intrinsic susceptibility. So like we were talking about, if there are specific pesticides that are being used in the environment, how broadly can we anticipate extrapolating that knowledge to other species? In the context of endangered species and derivation of aquatic life criteria, the tool has also been used and published. Most recently, we've started publishing case examples looking at bioaccumulation potential across species relative to perfluoroalkyl substances. The tool can be used for generating research hypotheses as well as for prioritization strategies. But I think Dries did a really nice job of talking about how we can utilize these types of approaches. And our group specifically has outlined how we can look at adverse outcome pathway networks that have been existing and specifically take out adverse outcome pathways and apply these bioinformatics approaches along with different types of systematic review methods to be able to bring in empirical data to support the taxonomic domain of applicability. So in thinking about an AOP network that was developed for honeybees, we demonstrated in the recent paper that is pending minor revisions for publication that looks at defining the taxonomic domain of applicability utilizing bioinformatics approaches such as SeqAPASS, as well as bringing in empirical data that exists to be able to define the domain of applicability. So as Dries had mentioned, we can utilize SeqAPASS to generate predictions of susceptibility that can define the biologically plausible taxonomic domain of applicability. So you'd have for molecular initiating events and key events that are early in the pathways where proteins have specifically been used to define the key event or the molecular initiating event. We can begin to extrapolate that knowledge from the species that are existing in the AOP from the developer and extrapolate that information to lists of hundreds of species, specifically being able to narrow that down to the biologically plausible taxonomic domain of applicability at the key event relationship level, and then across the entire adverse outcome pathway framework as we narrow down to the overlapping species amongst the key event relationships. We can then define the overall taxonomic domain of applicability for AOPs by determining those species that are in common across all key event relationships. And so this is a paper that will be coming out over the summer. And then to wrap up, I want to talk about, we've heard from multiple different groups across the globe. So, US as well as Canada, as well as the EU. And, one of the efforts that we've been leading is generating an international consortium to advance cross-species extrapolation and regulation. So ICASER. There's a number of us involved from government, industry, academia, as well as NGOs to bring together a group that can focus on defining taxonomic domain of applicability as far as having a greater understanding of the regulatory challenges globally and needs for cross-species extrapolation. And as a starting point, what we're doing is we're working on developing a bioinformatics toolbox using the expertise of many of us that are working to develop the tools, as well as champions within different agencies and across different organizations that are interested in utilizing these types of approaches for decision-making, and particularly in the context of regulatory decision-making. And then certainly being able to communicate that shared vision for how these types of tools can be utilized for decision-making after defining those challenges and those needs. So that is something that we're moving forward with a number of us. Here's the steering committee that's been working on generating the motivation behind the international consortium. And we're hoping that bringing together a global group to be able to focus on these types of issues that are already working, on these various topics within their own organizations will advance the science and be able to help us generate the criteria and the guidance for being able to utilize this information. So with that, I'm going to wrap up this talk just by acknowledging those at EPA and GDIT that have helped to develop the SeqAPASS tool, which was just released version 6 on the web. I'd invite everyone to go ahead and take a look at the tool. The URL is listed below, and always happy to help new users get familiar with the SeqAPASS tool and use that for their own needs in cross-species extrapolation.
Thank you so much, Carlie, for this very interesting talk and sharing all these resources available to regulators. So we are reaching the end of our webinar today. We are a bit in excess of the time we had anticipated. We have some messages in the chat box that we will certainly be using as much as possible to request or invite feedback from the WNT from this webinar today. I would like to thank you all, the speakers first, for very interesting presentations today. The recordings will soon be available to the WNT, and thank you to all of you in the WNT who could participate today in this webinar. I thank you so much. If you still want to post a couple of messages in the chat, please do so. I'm not closing it just at the minute. And have a great day, a great evening in Asia, Australia, and a good day elsewhere in the world. Bye.
Bye.